Hello everyone, I hope all of you are doing good. So today I am here with the second video of the video series on microorganisms friend or foe, class 8 science. And in this video we are specifically going to talk about the friendly microbes, the microbes which are in the friends category for us. Let's get started. So here are our friendly microbes. So several microbes are also useful to human beings in many different ways. Now for many it might be surprising that till now we only knew about germs, that microorganisms are germs, but basically germs and microorganisms are not synonyms. So the word germ is basically used for those microbes which cause diseases. But there are good microbes as well, which instead of causing diseases, they help us in many different ways. So how do microbes help us? What are some of the ways in which they help us? So they help us in a lot of household products. So they actually help in the preparation of these products like curd, cake, bread. Now I am sure that we consume all these in our day-to-day life. And in their preparation, microbes play the most important role. They also help in a lot of industrial products, like products which are prepared on a commercial scale in the industries, like beverages, antibiotics. So antibiotics, they are a boon to us. Now see what a contradiction. Antibiotics are used to get rid of diseases, and the diseases are also caused by microorganisms. So on one hand we have a group of harmful organisms which cause diseases. On the other hand we have a group of helpful microorganisms which help in preparing antibiotics, and these antibiotics will cure the diseases. So that means they serve both the purpose. They also help in preparation of a lot of enzymes and acids, and these enzymes and acids are again very much helpful in sewage treatment. Sewage treatment is another important application. So right now you might not know what exactly is sewage treatment. So I'll just quickly tell you what it is. What is sewage? Sewage is the unwanted water which has a lot of pollutants and which can cause air pollution and water pollution, and it comes out of your house every day. So this sewage, if you directly dispose them off into a river or a pond, what happens? The water in that river or pond gets completely polluted and the water becomes unsuitable for drinking purpose. So what needs to be done is this sewage should be properly treated before it is disposed of to any other water body. So how do we treat a sewage? A sewage is treated primary treatment and then followed by secondary treatment, in which all the harmful substances which are present in sewage get converted into less harmful substances, and then they are disposed of to the water bodies. So in that case the water bodies are not polluted to that extent. So this is basically sewage treatment, and in the process of converting more toxic substances into less toxic substances, microbes play a major role. Now we are not going to discuss about sewage treatment in detail right now, but if you want to understand it for your understanding, you can refer the video lessons of class 12th biology or class 11th biology, where I have explained sewage treatment in detail. Biogas production. So biogas is another environment friendly gas or a fuel, and in the production of this type of fuel also microorganisms play a major role. Control of pests and diseases, like the pests and diseases which happen in case of plants or in case of animals also, controlling them also microbes play a major role. They are also used as biofertilizers as well as decomposers. So what are biofertilizers and what are decomposers? So biofertilizers are living organisms or microorganisms which act as fertilizer. What do fertilizers do? In our previous lesson we saw that fertilizers improve the soil quality. They enrich the soil with more useful nutrients. So some microorganisms can help to enrich the soil with useful nutrients, and that is why those organisms are called biofertilizers. So that is one way it can help us. They can also act as decomposers, that is, they break down dead and decaying matter into useful inorganic substances. So that means it helps the environment to get rid of the unwanted dead and decaying matter. So these are some of the ways in which microbes can be helpful to us. With microbes in household. Now let us look at some of the household products where microbes play an important role. So one of these is curdling of milk. Like, how is curd prepared? Curd is actually prepared from milk. So how this conversion of milk takes place into curd, the major role is played by the microbes. So microbes are responsible for milk turning into curd. So curdling of milk is one such application. Baking, so all types of baking products, whether you talk about cake or you talk about bread, so there also microbes play a very important role. Cheese formation, we all often use cheese in our food items. So this cheese is also prepared by the action of microbes. Pickles. Pickles which are stored and preserved for a long time. So you would have seen your mother or your grandmother, they prepare pickle and you store it for a very long period of time. So these pickles also are prepared with the help of microbes. So these are some of the household items where microbes play an important role. So we are going to discuss about each of these and we will see that what role exactly is played by microbes in each of these products. So let us first talk about curd. So how is microbe playing the most important role in curd preparation? So what is curd actually? Curd contains a bacteria called lactobacillus. So this bacteria is always present in the curd. So next time whenever you eat curd or whenever you see curd, so always remember that there is a bacteria which is already present in the curd. But do you think that curd is poisonous? Not at all. In fact, curd is something which is extremely healthy for us and we eat it. So what happens? Once we eat curd, do we fall ill? No, but the bacteria is already there. So the bacteria which is present inside the curd is a friendly bacteria and it is not a harmful bacteria. That is why it doesn't harm us. So now what we have to see is how exactly this lactobacillus help in the process of conversion of milk into curd. So lactobacillus, it is a friendly bacteria as I said, and it treats and prevents diarrhea. Now forget about the fact that it is going to harm us. It is going to help us. It helps us to prevent diarrhea, and that is why you would have seen that when you are suffering from diarrhea, it is prescribed or you are advised to eat curd because curd contains this bacteria and it will prevent or it will treat the problem of diarrhea. It also helps in food breakdown. Now once you eat the curd, this bacteria gets inside your body. So this bacteria helps in an indirect way in the process of digestion, because in the digestion process what happens, the complex food particles are broken down into simpler forms. So this bacteria also plays an important role in the process of food breakdown. It also helps in absorption of nutrients. Now once the food is digested inside our body, then the food is absorbed by different parts of the body, and that's how we get energy out of the food. So this bacteria also helps in absorption of nutrients. So you see, this bacteria after getting inside our body, they help us in a number of ways. So now let us see how exactly the process of curdling takes place. That is conversion of milk into curd. This process is called curdling. So how this process actually happens. Now, have you ever seen your mother preparing curd, or have you ever yourself tried to prepare curd? If not, you can try it at your home. So all you need is milk and a little bit of curd, maybe a spoon of curd. That should be enough. Okay. So what you have to do is you take a bowl, put some milk in that bowl. Now that milk should not be boiling hot and it should not be freezing as well. So it should be little warm. So you take milk in that bowl and you add a small amount of curd into that milk, and then you leave it overnight. Next morning when you see that it has transformed into curd. So basically that small amount of curd is able to convert that bowl of milk into a bowl of curd. So what exactly happens? Now as I said, this spoon of curd, even though it is very small amount of curd, but that small amount also contains the bacteria lactobacillus. So what this lactobacillus does, that is what we have to concentrate here. Now even though the amount of curd which is added is very small, but this small amount of curd contains millions of lactobacillus. And what do these lactobacillus do? These lactobacillus, they convert sugars into lactic acid. And where are sugars present? Now milk. So here you have milk, and milk contains a sugar called, what is the sugar which is present in milk? The sugar which is present in milk is lactose. So milk contains lactose sugar. So the word lacto is related to milk. Therefore, the sugar present in milk is lactose. So this lactobacillus will convert lactose into lactic acid. So basically it will convert sugar into acid. So lactic acid will be formed. Now what happens when lactic acid forms? This lactic acid will coagulate and partially digest the milk protein. Now this milk, the way milk is, it is because of the presence of the various particles in it. Now there is a milk protein. Now the protein gets destroyed, the structure of the protein gets altered, and this alteration happens due to the formation of lactic acid. Now what happens? This lactic acid, due to the formation of this acid, it imparts a sour taste to the curd. So that is why there is a big difference between the taste of milk and the taste of curd. So curd is sour in taste, but when you taste milk, it is kind of sweet, right? So you can say that milk is kind of sweet. So milk is sweet due to the presence of sugar, and curd is sour due to the presence of acid. And who converts sugar into acid? Lactobacillus. That is why for curdling to take place, it is extremely important to add a small amount of curd. If you do not add that, you are basically not adding the lactobacillus bacteria. And as long as you do not add this bacteria, the sugars will not get converted into acid, and milk will also not turn into curd. So what happens is, at a suitable temperature, like normally the room temperature, these lactobacillus, they start to grow and multiply. Now as lactobacillus grow and multiply, what happens? More and more coagulation of proteins take place. As I said, the milk proteins, they start coagulating. Therefore, the nutritional quality is also improved. For example, if you compare the nutritional quality of milk with that of curd, you will see that in curd, the content of vitamin B12 is much more than that of milk. So all these changes happen. So who is responsible for this entire change? Lactobacillus. And lactobacillus is a bacteria. So here you can see that this bacteria is not our enemy. Instead, this bacteria is our friend. It helps in preparing a better nutritional product like curd. Even when it gets inside our body, it helps in breakdown of food. It helps in absorption of nutrients. It helps to treat diarrhea. So there are a number of ways in which this bacteria help us. So this is one example of a friendly bacteria. So as I said, the increased acidity causes the milk proteins to turn into solid masses. So that is why your milk is liquid, but when you look at the curd is not liquid. So curd is like solid masses. So this conversion happens due to the coagulation of the milk proteins. Which milk protein is present? The milk protein is casein. Now let us see how microbes play an important role in the baking industry. So, have you ever observed that if you take some maida, that is flour, and you add, normally the way you prepare a dough, you try to prepare a dough, all you do is just add a small amount of yeast to that dough. So, this is how you prepare the dough. And leave it for a couple of hours, maybe some 2 to 3 hours. And after 2 to 3 hours, you will see that the dough actually rises like this. So what helps the dough to get fluffy, or what is the rising agent which you have added to this dough? So this rising agent is nothing but the yeast. So yeast acts as the rising agent. And what is yeast? Yeast is an example of a fungi. So basically microorganisms help you to act as rising agent, and that's how it helps to bake a lot of items. So let us see how it does. So yeast is used as a raising agent in baking, and in baking you cannot do it without a raising agent. You would have seen, like when you prepare a cake or when you look at the bread, so it is very fluffy. So inside it is not very solid. So gaps are created in between, and that's how it has been made very fluffy. So in order to make that fluffy, you need a raising agent, and that raising agent is yeast. And that's how you prepare cakes, muffins, and all these bakery items. So the most common yeast which is used for baking purposes is Saccharomyces cerevisiae. So this is the most commonly used yeast. So let us see how yeast helps in baking. Now what yeast does is it converts the carbohydrates in the dough into carbon dioxide. So this is the conversion that takes place. So carbohydrate is present in the dough because the dough is made up of flour. So flour has carbohydrates. So this carbohydrates get converted into carbon dioxide. Now what happens due to the formation of carbon dioxide? Due to carbon dioxide, the dough expand or the dough rise and it forms bubbles. So carbon dioxide, which is a gas, it leads to the formation of bubbles, and that is why you will see that when you leave this dough for a long time, and after that when you see it, you will see, first of all, it is raised, and also there are bubble-like structures, tiny bubble-like structures which is formed due to the formation of carbon dioxide. Now once the dough is baked, for example, you prepare the dough, you leave it for some time and then you bake it. Now once you have baked it, what happens to the yeast? Because yeast is a living organism. So the yeast dies once it is baked. Now what is the result? The result is that the dough is already expanded. So there are bubbles in the dough. So once it is baked, it becomes very soft and spongy and the yeast dies. So the yeast kind of sacrifices its life to prepare that cake for you. So soft and spongy baked product is formed. Now many a times if you look at the recipe of a cake or any other bakery item like bread, you will see that use of eggs and sugar. Either good amount of sugar or eggs are used because these substances accelerate the growth of yeast. They favor more and more growth of yeast. So you have more amount of yeast, so you get more fluffier cake. So the baking is better when you have more yeasts. So now in a very similar way, not only for cakes, in bread making also similar type of process happens. So initially aerobic respiration takes place. Like how exactly this conversion happens? For this conversion to take place, aerobic respiration takes place. That means the yeast, that is the fungi, they undergo aerobic respiration, that is in presence of oxygen. So they utilize oxygen and they give out carbon dioxide. So as a result of this aerobic respiration, what happens? Carbon dioxide and water, they are formed. So in this process, what happens? Oxygen is used up. Oxygen will be used up and carbon dioxide and water will be formed. Due to the formation of this carbon dioxide, the rising happens or the expansion of the dough happens. Now what happens when all the oxygen has been used up by the yeast? Once all the oxygen is used up, then anaerobic respiration happens. That is, fermentation takes place. Now when all oxygen is being used up, in that case anaerobic respiration will take place, and as a result, in this case, ethanol will be formed. So let us not get into all these details because you will be studying about all these in your higher classes. So for now you should understand that yeast, it converts carbohydrates into carbon dioxide, and the process which helps in this conversion is the aerobic respiration, that is oxidation in presence of oxygen. Now this carbon dioxide rises the dough, or it helps to expand the dough, or it helps in the bubble formation. Now once the cake is baked, the yeast dies, and due to the carbon dioxide, a soft and spongy baked product is formed. So now let us look at the dough making process for various other food items like idli and dosa. Now you would have had these as your favorite breakfast items when you have idli or dosa. So how do you prepare them? So for this also you would have seen that your mother prepares the dough and leaves it overnight, leaves it to get fermented. So what happens here? So in this case also there is a bacteria called lactic acid bacteria which ferment the idli or dosa batter, because in this batter you do not use any yeast. neither you use any curd, so you really don't have anything which can act as a rising agent. So what is being used here? So here the microorganism which play an important role is the lactic acid bacteria. So where is this bacteria present? Now how do you prepare the dough for idli or dosa? So for that purpose you use rice paste as well as a specific type of pulse called the urad dal. Now these bacteria, that is lactic acid bacteria, they are naturally present in the urad seeds, and this is the specific type of dal which you use to prepare idli or dosa. So now due to the presence of this type of bacteria, when you leave the batter for quite some time, then these bacteria will lead to the formation of carbon dioxide. So as a result of what is the process in which carbon dioxide will be formed? The process is called fermentation. So fermentation happens in absence of oxygen, and the result is that carbon dioxide will be formed along with lactic acid. So carbon dioxide and lactic acid will be formed. Due to the formation of lactic acid, it gives a slightly sour taste. So formation of acid will give a sour taste. So if you see, the idli batter or the dosa batter has slightly a sour taste. So that is due to the formation of the lactic acid, and due to the formation of carbon dioxide, it again, the dough again rises. So that formation of bubble happens due to this formation of carbon dioxide. Now you would have observed that if idli batter or the dosa batter is fermented for a very long time, like instead of a couple of hours, if you leave it for a couple of days or so, what happens? Then the batter becomes too sour. Now it becomes too sour due to accumulation of too much lactic acid, because the longer the duration, the more is the formation of lactic acid, and more is the formation of lactic acid, more is the sour taste. So gradually it will turn too sour. Therefore the batter should be fermented only for a limited period of time. In this case, if you see, during the dough preparation, you do not need to add any starter here. So you neither need to add yeast, you don't need to add any other thing, because the bacteria is naturally present in the urad seeds. Now these kind of examples can also be seen in other food items preparation. For example, the bhature which we prepare when we prepare chole bhature, or the naan which we prepare, where the kneaded dough is kept aside for some time for a rising, so that the process of fermentation can take place. So in many of them we add baking powder, in many of them we add baking soda, in some of them we add yeast powder. So in preparing different things, we add different types of agent which can make the fermentation successful. Now you would have seen that some people use baking powder, some people use baking soda, some use yeast. So why do we use different things or different agents for the same process? That's because each of them have its own advantages and disadvantages. For example, if you talk about baking powder, so baking powder can be stored for a very long period of time. So baking powder or baking soda, they can be good alternatives to yeast. They also function as yeast, that is, they can act as raising agent. Now, keeping or preserving yeast for a very long period of time is not a good option, because they do not last for a very long time. So that means baking powder, baking soda are good alternatives to yeast. So this is how in our household items we see that microorganisms play an important role. So now let us look at the role of microbes in industrial products. Now, as I had mentioned before, that there are quite a few things in terms of the commercial products which are produced on a commercial scale in the industries. There are quite a few things in which microbes play an important role. Some of them are antibiotics and beverages. Antibiotics, they are extremely helpful because they help to cure so many diseases. Beverages, again, a lot of different types of beverages like wine, beer, whiskey. Preparing all those things also need the role of microorganisms. So first, let us discuss about antibiotics. Now, you would have had antibiotics when you have fallen ill. The doctor gives you a course of antibiotic, a set of medicines which he prescribes to take for a particular number of days, maybe a 3 days course. So even if your fever abates in a day or two, but you still need to continue taking the medicine for the prescribed number of days. For example, if the doctor had asked you to take it for 3 days, you will have to take it for 3 days. So if you look at the word antibiotics, so what does it mean? Anti means against. So anti means against, and biotics. So bio means something related to life. So you mean the word means against life. Now that doesn't mean that it is something which is against life and it is going to kill us. Instead, it is just the opposite. What it means is it is against bacteria, that is against the living organisms which cause diseases. So that ways it is against life, and that is why these antibiotics are against the bacterial infection. So these are chemical substances which are produced by microbes, and they kill disease causing microbes. So if you see here, antibiotics are the best example where we can see both the positive as well as negative sides of the microbes. So these antibiotics are produced by microbes, and what do? They kill other microbes which are harmful. So antibiotics is produced due to the good microbes and it kill the bad microbes. So that is how antibiotics are. So they treat bacterial infection. Now antibiotics are very specific in their action. They can only treat infections which are caused by bacteria. So antibiotics are not useful against viral infection. So you would have observed that when you get fever, sometimes you get fever due to some sort of bacterial infection, maybe some throat infection or some other infection caused by bacteria. So at that time your doctor will prescribe you a course of antibiotic. But if you get a viral infection, that is due to viral infection you got a viral cold or flu, the doctors will not give you antibiotics. So antibiotics can only treat bacterial infections. They will have no effect on the viral infections. So this antibiotic was discovered for the first time by Alexander Fleming. He was a Scottish scientist, and in fact you will be surprised to know that he discovered antibiotic by chance. So he was not performing an experiment to discover antibiotic. So he was doing some other experiment when he accidentally discovered the first antibiotic, and the first antibiotic which he discovered was named as penicillin. So penicillin was the first antibiotic that was discovered. Now any discovery which happens by chance is known as serendipity. So serendipity. So one example of serendipity is the discovery of penicillin. So serendipity means discovery by chance. So you are not performing your experiments to discover this, but by chance you discovered it. So that is serendipity. So now let us spend some time trying to understand how exactly was penicillin discovered. So because that is an interesting story. So discovery of penicillin. Now as I said that penicillin discovery was a chance discovery, which is often called serendipity. So Fleming was experimenting with Staphylococcus bacteria. So Staphylococcus is a particular type of bacteria. Coccus are those bacteria which are spherical in shape. So he was experimenting with this particular type of bacteria. Now what happened was, it was observed that a particular petri dish, so this is called a petri dish, the dish on which the culture is grown, bacterial culture is grown and for experimentation purpose. So once what happened was, a petri dish was left open by mistake. Like when he went back home after his work, he forgot to close the petri dish. So it was just left open by mistake. And when he came back later, he observed that the petri dish was contaminated by blue-green mold. So this is the Staphylococcus bacterial growth. This is what he was studying in this petri dish. Now, when he kept it open for some time, and that too by mistake. He did not purposely leave it open, but later he found that there was some mold growth on one side of the petri dish. So this was his observation, and this mold growth was quite visible, and The most important thing that he observed was that this mold contamination inhibited the bacterial growth. So wherever this mold growth had taken place, there was no bacterial growth taking place in that part. So this proved that mold inhibited the growth of the bacteria, right? So what did he get to know out of this? So he got to know that mold was something which was stopping the bacteria to grow. So now whenever we get bacterial infection, why the bacterial infection exists or why it spreads? Because the bacteria continuously keep on growing, and bacteria, they multiply really fast. So that means in order to treat bacterial infection also, we need something which can inhibit bacterial growth. So that is what he could see here. On this cell culture dish, he could actually find some organisms which could inhibit the growth of the Staphylococcus bacteria. So what he concluded was mold released a substance that inhibited bacterial growth. So, and what was mold? Mold was nothing but a fungi. Mold is an example of fungi. So, this fungi was actually releasing some substance which inhibited or which stopped the bacteria from growing. So, penicillin was named after this mold, Penicillium notatum. So, this mold was Penicillium notatum, and after that the antibiotic was named as penicillin. So here if you see, the medicine was prepared from this mold, that is antibiotic was prepared from the mold which is a microorganism, which is a microbe, and this microbe is going to kill another microbe which can be harmful. So that is what antibiotic does. So this is how the first antibiotic penicillin was discovered, and you see how this discovery happened just by chance. Now, microbes, when we talk about the medicinal uses of microbes, we also see that they play a very, very important role in the process of vaccination. And I am sure all of you must be knowing how important vaccination is. Now, before we talk about the role of microbes in vaccination, let us quickly understand what is vaccination. You would have often heard small children, they go to the doctors to take their vaccine drops. So the polio drops or the vaccinations, in the form of injections. So you would have seen that they have some specific interval, maybe at 3 months, again at 6 months, 9 months, and so on. So what are these injections and why do they take it? So now our little friend is very happy to know that microbes also help to cure us, like how we studied about antibiotics. So now we are also going to talk about the vaccination process, where we will see that they help us even better. So in antibiotics, they cure a disease, and in vaccination, they also prevent a disease. So let us see how. So what is vaccination? Now this is the picture of a vaccination camp where small kids are given the vaccine drops. So it is a process in which vaccine is given to improve the immunity of the body against a specific disease. So quite a few terms need to be explained. What is vaccine? What is immunity? Now let us first talk about immunity. What do we mean by immunity? Immunity is our body's own defense system. For example, in order to protect our house, what do? We employ security guards outside our house who can take care of the security of the house. Correct? So in a very similar way, our body also has a security system inside which helps to protect the body against any sort of infections. So that is called the immunity of the body. If a person has a very strong immunity, in that case, he is less prone to get infected. Because he is strong, he has a strong immune system, so his immune system can fight well with the infection. If a person has a very weak immune system, he is more prone to fall ill. He is more prone to get attacked by some harmful microorganism. So now in the process of vaccination, what happens is vaccine is introduced inside the body of a person. And what is that vaccine? So that is where we will see that microorganisms play a role. So vaccine will be introduced inside the body, and the purpose of this vaccine is to improve the immunity of the body against a specific disease. For example, if polio drops are given to kids, that means the immunity of the body against the polio disease is being developed, so that if the polio disease, if the microbe which causes the polio disease that attacks that person, the body will be able to fight against that microbe and the body will not get polio. Why? The reason is the body has developed a very strong immunity against polio disease. Now, how the body developed that immunity? Due to the introduction of the vaccine. So, what is this vaccine? Vaccine is a biological preparation that resembles a disease-causing microbe. Now, it is something like this. Let us suppose your body has encountered a particular microbe once. Now, inside our body also, inside our immune system, we have certain different types of cells. Now, there are certain types of cells which keeps the memory, which keeps a record of the memory. So now, once your body has been attacked by a particular type of disease causing microbe, so your body remembers that. So it is there stored in the memory. So the next time when the same microbe comes and tries to attack, so the body tries to fight back with greater immunity so that the disease cannot win over and the person is not affected by that disease. So that is how the system of our body works. So in vaccination, what happens is a false indication is given to the body that a disease-causing microbe has attacked the body. Now, what is that disease-causing microbe? Or what is that biological preparation? For example, a particular disease is caused by a particular microbe. Now, we do not want a person to suffer from that disease. So, we cannot directly enter that microbe. But how do we make sure that the body should realize that the microbe has entered? So this vaccine is made up of dead or very weak microbes. So in this case, what will happen? If a very weak microbe is entering inside your body, you are very less likely to attack that disease. You will not get that disease because the microbe is very weak or the microbe is dead. So that microorganism is dead. So let us suppose a particular disease is caused by a virus. Now what I do, instead of introducing that virus into the body, I introduce the same virus which is dead. So now it is very obvious that you will not get that disease, but at the same time, your immune system will get to recognize or it will store that particular microbe in its memory. Even though it is dead, but it will store that microbe in its memory. So next time, if a live microbe attacks your body, your immune system will fight back with greater strength, and that's how it will not allow that microbe to win over and cause the disease. So that is the concept of vaccination. Now in vaccination, where is the role of microorganism in the preparation of vaccine? Because to prepare vaccine, you need samples of microbes. You need microorganisms which are either dead or which are very weak. So that means these kind of microorganisms help in the preparation of vaccine, and the vaccination has been a very successful process because it has been able to eradicate a lot of deadly diseases. So let us try to understand the concept of vaccination, because the concept might sound a little tricky to you right now, but I will try to give you an example which will help you to understand the concept very easily. Now forget about disease, forget about microorganism, forget about the body. Just look at this example. That how a creation of memory happens with the help of vaccine. So vaccination is all about creation of a memory of a disease causing organism, and that is why we introduce a dead virus or a dead bacteria into the body so that the body stores a memory of that particular virus or that particular bacteria, so that when the same virus or the same bacteria attacks the body again, the body will not allow that bacteria or virus to enter or to cause that disease. So let us take this example. Let us suppose this is a palace, or this is your house, let us assume. Okay, so you want your house to be secured. When I say secure, you want it to be well protected from robbers and dacoits and thieves. So that is why what did you do? You employed one security guard outside your house because you want it to be completely protected. Now what happens? Let us suppose a thief comes in. So when this thief tries to enter inside this house, this security guard who was standing outside to ensure the security of the house, he gets to know that in order to, he gets to recognize this person or this thief. He gets to know that, okay, fine. So these type of thieves might come and they might try to enter inside the house. But now this time, the thief who has come, he has come without much preparation. Right? So he has just come thinking that, okay, there is just one security guard, so let me try my luck. So that's how he came. So now when the security guard got to know that, okay, this house is very prone to attack by thieves. So what he does, he increases his power. So he increases his immunity. So now what happens is, the next time when the thief even thinks of coming near this house, he gets to see that the immunity or the protection system is very strong. So the immune system of the body is very strong now. So what happens, as soon as the thief tries to come near the house, all these group of soldiers, they fight back and send him away. So the same concept is applicable for vaccination as well. So when you apply the vaccine, you are actually sending a very weak microbe which is not capable of causing the disease. But that microbe is capable of creating a memory inside your body. So the same thing happens in case of vaccination. Now when the same virus, now let us suppose if the real virus attacks your body, the real virus or the real bacteria which is capable of causing a disease, if that microbe attacks your body, now your immune system already has the memory of that particular virus or that particular bacteria. So the body will fight back against that microbe with greater strength, and therefore it will send the microbe away. So the microbe will not be able to cause the disease. So this is how the concept of vaccination works. So I hope now you understood where is the role of microorganisms here. The vaccine or the sample which is prepared to create memory inside the body, that is prepared with the help of weak microbes or dead microbes. So that is where microbes play a role in the process of vaccination. Now, what exactly happens during vaccination? Now, as I said, memory gets created and then the same memory is used up in the next time. So, how exactly is that memory created, and what happens when next time the same virus attacks the body? So here comes the concept of antigen and antibody. Now these might be a little complicated for you guys, but this is very interesting and that's why I feel that you should know it. So let us see, what are these? Now what happens is, whenever a vaccine is introduced into the body, that is a new microbe is being introduced into the body. Either the microbe is weak or it is dead, doesn't matter. But a new microbe is being introduced into the body. So the body produces antibodies in response to any new thing which is introduced into the body. So here the vaccine is actually introducing a foreign particle inside the body. So this foreign particle is called antigen. And the antibodies are always produced in response to the antigen. And these antibodies which are produced inside our body, they remain inside our body and they will fight any further attack of the same antigen. You got it? So whenever a particular virus enters your body for the first time, so anything which enters your body, that is called as antigen. So when it enters for the first time, so the body by default produces antibodies against that antigen. Okay? These antibodies will remain inside your body. It will sit inside your body silently. But next time if the same antigen attacks your body, so the antibodies will fight back. So that is what happens during vaccination. So let us try to understand the terms antigen and antibodies even better, because you might ask, what are these? I am hearing it for the first time. So in simple words, you can just think it in this way. The thief which we were talking about is the antigen, and this security guard is nothing but the antibody. So when the thief was trying to enter inside the house, so the house created the antibodies, or the house had the soldiers, or the house had the security guards. So in response to the thief, now as long as the thief did not come, so do you think that the security guards were fighting with guns with somebody else? No, not really. But as soon as the antigen came in, whenever a foreign particle came inside the house, these antibodies were produced. That is, they started fighting. So antigens are the foreign substances and antibodies are produced inside the body in response to the antigens. So to make it more detailed, antigen refer to any foreign substance inside the body. Now this foreign substance can be anything. It can be chemicals, it can be microorganisms like bacteria, virus, it can be toxic materials, that is poisonous materials, it can be pollen, so it can be anything. Any of these is called an antigen. So any foreign substance which is entering your body for the first time, that is an antigen to the body. So these antigens trigger immune response to produce antibodies. So antibodies are created due to antigens. So antibodies are kind of response to antigens, right? So what are antibodies? So these are protein structures which are created inside our body. So they have a specific shape and they have the shape of the English alphabet Y. So this is how an antibody looks like. So you see, it has a Y-shaped structure, and if you see here at one end they have antigen binding site. So if you see, this is antigen. So here you can see all these are examples of antigen. So this is how antigen looks like. Now whenever an antigen enters inside your body will produce Y-shaped protein molecules such that this antibody will bind to this antigen. So you see, this portion of the antigen fits exactly to the antigen binding site of the antibody. So basically they both fit together perfectly. So now what will happen? This antibody, this Y-shaped structure will remain inside the body forever. Now next time when the same antigen comes in, what will happen? So the same antigen will fit inside the antibody and the antibody will destroy the antigen. So the antigen will not be able to cause any kind of disease inside the body. So that is how the concept of antigen-antibody works. So these antibodies, they recognize and neutralize the pathogens. Pathogens are nothing but the antigens. So each antibody binds to a specific antigen. So if you see here, now let us suppose if this antibody is produced inside your body. Now if this antigen attacks the next time, so this antibody will not be able to protect your body against this antigen because this antibody cannot bind to this antigen. So one specific antibody can bind to one specific antigen. So it can give you protection against that particular antigen. So that is how it works. Now, if you talk about the structure of an antibody molecule, you can see here it is a protein molecule. It has peptide chains. There are 2 light chains and 2 heavy chains. So this is a light chain. This is also a light chain. And this one is a heavy chain. This one is another heavy chain. So these are the heavy chains. So there are 2 light chains and 2 heavy chains. So that is how the structure of antibody looks like. So I hope the concept of vaccination is clear to you because this is very important to understand how vaccination happens and how microorganisms help us in the process of vaccination. Now let us quickly look at the success of vaccination. As I was telling you that it has been a huge success because many deadly diseases like smallpox have been eradicated only because of vaccination. So smallpox is eradicated because a lot of people died because of this disease because there was no treatment. So vaccination could actually prevent this disease to such a large extent that now nobody suffers from smallpox. Diseases like polio, tetanus, measles have been reduced because vaccines are available for each of these diseases. Now it is a parent's responsibility to get these vaccines on correct time to their kids so that they do not suffer from any of these. So now let us talk about the next useful application of microorganisms, that is in the beverages. Now as I was telling that it also helps in the preparation of alcohol. So let us see how microorganisms will help us to prepare alcohol. Yeasts play an important role in the production of various alcoholic drinks. So when you talk about alcoholic drinks, it could be beer, whiskey. So there are many different types of drinks which are available. And yeasts again play a very important role in their preparation. So let us see how yeasts play a role there. So commonly the yeast which is used for this purpose is Saccharomyces cerevisiae. Now, as I was telling before also, this yeast is also used as a raising agent in most of the bakery items. So this yeast is often called as the brewer's yeast because of its application in preparation of alcohol and as a raising agent. So it is often termed as the brewer's yeast. So let us see how the process of alcohol formation takes place. So let us now learn about the process of alcoholic fermentation. So this process is also known as ethanol fermentation. That is because the product here is ethanol. And what is ethanol? It is C2H5OH. Ethanol is an alcohol. And in this process, ethanol is produced. And that is how alcohol is produced. So what happens in this process? The yeast Saccharomyces cerevisiae, it ferments the fruit juices and malted cereals. So basically, what will the fruit juice or the malted cereal made up of? So the malted cereal will contain the sugar called maltose. Similarly, if you talk about the fruit juices, they will contain fructose, sucrose. So these kind of molecules like fructose or sucrose or maltose, all these are sugars. So all these sugar molecules, they get converted into ethanol and carbon dioxide, and a lot of energy is released during the process. So what happens is yeast perform this process or this conversion in absence of oxygen, and that is why it is called fermentation. So fermentation always happens. Fermentation is basically anaerobic respiration, and it always happens in absence of oxygen, whereas aerobic respiration happens in presence of oxygen. Now in fermentation, what will happen? All these sugar molecules, whether it is fructose or maltose or sucrose or lactose or any kind of sugar molecules, they are either disaccharide or polysaccharides. So end of the day, they all are made up of simple sugar, and simple sugar is glucose. So C6H12O6, this is glucose. So sugar gets converted into ethanol, that is C2H5OH plus carbon dioxide. So this is how the balanced reaction will be. And this entire conversion happens in presence of an enzyme called zymase. So this enzyme, what it does, it actually helps in the conversion of this reaction. And this entire process, who makes this entire process possible? This entire process is made possible by the yeast, Saccharomyces cerevisiae. So now this carbon dioxide which is produced here, this production of carbon dioxide helps this yeast to act as a raising agent, because the carbon dioxide helps in the formation of bubbles and it also raises the dough. So that's how this yeast is used as a raising agent. And this ethanol which is produced here helps in the preparation of alcohol. So it is the same process where both the products are formed, alcohol as well as carbon dioxide. So this is how alcohol fermentation takes place. Now, this alcoholic fermentation also takes place on a commercial scale. So, when we want to commercially produce a lot of alcohol, so that cannot happen in a small laboratory. So, for that, fermentation plants have been set up. So, in the picture you can see, this is how a fermentation plant looks like. So here fermenters are used for large scale production of microbes, because you need large amount of microbes as well. Little amount of yeast will not help you to produce a lot of alcohol. So the greater amount of alcohol you want is the greater amount of yeast that you will have to provide. So in order to produce so much of yeast or so much of microbes, you need fermenters which can produce so much of microbes. So this is how fermenters look like. So they are like big tanks inside which a lot of microbes can be grown, and then these microbes help in the conversion of sugars into alcohol. So you can carry out this small experiment on fermentation in your school laboratory if the facilities are provided there. So what you can do is, you can actually see the process of conversion of sugar into alcohol. This is called fermentation, and it happens in absence of oxygen. So normally the word anaerobic respiration and fermentation are used interchangeably because theoretically it refers to absence of oxygen. However, in this simple experiment, it is not that you have to create anaerobic conditions. Even in presence of oxygen, you can see how sugar gets converted into alcohol. So what you do, you take a beaker and take some water in it. Now you add some amount of sugar into the water. Now what you do? Now you add some amount of yeast into the sugar solution. So now you have yeast, you also have sugar. So what we know in fermentation, yeast will convert sugar into alcohol. Now as soon as you added the yeast, now you give some time for fermentation to take place. For that purpose, you cover it with a lid and leave it undisturbed for quite some time. Now, after some time, what you will see is the entire solution, the sugar solution which was white in color, gradually turns reddish. That is due to the formation of ethanol, that is alcohol. So this is how you can see how sugar gets converted into alcohol by yeast. So not only antibiotics and the alcohol, there are many other chemicals which are useful to us in different ways, which are prepared by microorganisms. Now several chemicals are produced by microbes. For example, organic acids, enzymes, alcohol. So these are some of the chemicals which are produced by microbes. So now let us quickly see some of the microbes which produce organic acids. We have already looked at some of the acids which are produced by microbes. For example, lactic acid, acetic acid. So let us see. For example, lactobacillus, which is present in curd, it helps in producing lactic acid. That's what it does. It converts the lactose sugar present in milk into lactic acid. And this lactic acid makes the milk turn sour, and that's why curd is sour. So this is one microbe which produces an acid. Acetobacter aceti, this is another microbe which produces acetic acid. So this is a bacteria which produces acetic acid. What is acetic acid? It is nothing but vinegar. So I hope all of you now understand what is vinegar. Clostridium botulism, this is another bacteria which produces butyric acid. Aspergillus niger, it produces citric acid which is commonly found in lemons and the citrus fruits. So citric acid can be produced by this fungi, Aspergillus niger. So these are some of the microbes which can produce organic acids. Now let us look at some microbes which produce alcohol. Just now we spoke about alcoholic fermentation, right? So we saw there that yeast produces alcohol. So the yeast which is used for alcoholic fermentation is Saccharomyces cerevisiae, which is also known as the brewer's yeast because it is also used for baking purpose and also for alcoholic preparation. So this yeast, it produces ethanol, which is C2H5OH, and during this process it also produces carbon dioxide, and due to the production of carbon dioxide, this yeast acts as a raising agent in bakery industry. So now we will see how microbes are useful as biofertilizers. Now before we talk about microbes used as biofertilizers, first let us try to understand what are fertilizers and why do we need them. Now fertilizers are those substances which are added to enhance the fertility of the soil. Now if we want to grow crops, if we want to increase the crop productivity, we need to ensure that the soil is fertile enough so that the plants which grow on that particular soil can get all the required nutrients, minerals, and thus can give better productivity. Now adding fertilizer is just like externally adding more and more nutrients to the soil. So more phosphates and more nitrogen can be added to the soil by fertilizers. So these fertilizers result in higher yields and healthy plants because they directly increase the nutrient content of the soil. Now, there are certain disadvantages associated with chemical fertilizers. Now, some of these are The chemical fertilizers are basically chemicals, so they are basically made up of harmful chemicals which can be harmful for other life forms. Even though it is increasing the soil fertility, but it can be harmful for so many living organisms present inside the soil. For example, earthworms, other small insects which live in the soil, they might be harmed if this toxic material is present inside the crops, and if the crops are eaten up by other animals, it can cause harm in them. It can cause imbalance in the soil pH, so the acidity or basic nature of the soil can get disturbed due to the presence of lot of chemicals. Results in soil infertility. Though the purpose of fertilizer is to increase the soil fertility, but if fertilizer is applied to the soil in excess, in that case it can spoil the soil fertility, because too much of chemical substances in the soil will also spoil the nature of the soil. Degrades ecosystem, because when it is a toxic material, it can kill many life forms of the soil ecosystem. Therefore, the soil ecosystem might get disturbed. Plants become susceptible to many diseases. Sometimes what happens is, if the same type of fertilizer is being applied over and again very frequently and in high amount, so sometimes the plant become very much used to, or those kind of fertilizers do not work for the plants. So the plants, instead of growing better, they tend to get diseases. Fruits and vegetables have high toxic residues, as I said. Now, since these are toxic materials, there are chances that they might remain somewhere inside a fruit or a vegetable. So when that fruit or vegetable is being eaten by some other animal, it can cause some harm to that animal. Environmental pollution. If fertilizer is used in excess and immediately after fertilization, if the field is watered either by rain or by irrigation, so there are chances that the fertilizers might get washed away by the water, and it can cause water pollution to the nearby water bodies. Maybe the nearby pond or the nearby river might get polluted because of this toxic substance which might get washed away by water. So these are some of the disadvantages associated with the use of chemical fertilizers, and all these disadvantages are because of the poisonous nature of these chemicals. So we can say that the fertilizers are advantageous in terms of that it increases the fertility of the soil, but at the same time it is not eco-friendly, so it harms the environment at the same time. So therefore was introduced biofertilizers. So biofertilizers are like living organisms which act as fertilizers. So these are basically living organisms. So they are not chemicals. So they will not cause any harm to the environment. So they will be eco-friendly. But at the same time, they will act as fertilizers. That is, they will provide nutrients to the soil. So they will enrich the soil fertility. So that means it will have all the advantages of fertilizers, but it will not have the disadvantages of fertilizers. So these are living organisms which enrich the soil nutrient quality. Now, source of biofertilizers are bacteria, fungi, and cyanobacteria. All these microbes, they help to increase the nutrient quality of the soil. So let us look at some of the examples of biofertilizers. Now, one good example is the nitrogen-fixing rhizobium bacteria. So there is this bacteria called rhizobium, and where do we find it? We find it in the root nodules of leguminous plants. Now, what are leguminous plants? Plants like beans, peas, they all fall under the category of leguminous plants. So if you look at the roots of these plants, you actually see that there are these kind of structure, and because of these nodule-like structures. So in these root nodules are present these bacteria called rhizobium. So this is a symbiotic association between the rhizobium bacteria and the root nodules of leguminous plant. Now, what is symbiotic association? It is basically an association where both the parties get benefited. So one party is rhizobium, which gets its shelter and food from the root nodules, and the other party is this root nodule of the plant. So how is the plant getting benefited? Because this bacteria helps to fix atmospheric nitrogen into the soil. So now, if the soil has more nitrogen, who will take the benefit out of it? The plant, of course. So the plant allows rhizobium to stay in its root nodules because this bacteria brings nitrogen to the plant, and nitrogen is a very important nutrient for the plant. So the plant needs it. At the same time, the bacteria's advantage is that it gets its food and shelter from the plant. So this bacteria fixes atmospheric nitrogen, and therefore nitrogen being an essential nutrient for the plant, so we can say that it increases the nitrogen content in the soil. So basically this rhizobium bacteria acts as a biofertilizer because it is increasing the nutrient content or the nutrient quality of the soil. So this was one example. Let us look at the next example. It is again a symbiotic association. So let me write it here. Symbiotic association is an association where there is mutual benefit. So here again, this is another symbiotic association between fungi and certain plants. So this type of association is called mycorrhiza or mycorrhiza. So this is the name given to this type of association between fungi and plants. Now how the fungi helps here? Here in this picture you can actually look at the mycorrhizal root tips. So here you can see they have nodule-like structures, right? Their root tips. So here some fungi are present, and these fungi, they absorb phosphorus from the soil and pass it to the plant. So that ways it helps to increase. Again, they act as biofertilizers. How? Because they help to increase the nutrient quality of the soil, and it helps in the passage of phosphorus from soil into the plant. So again, another symbiotic association. So this is resistant to pathogens. Also, it prevents the attack of disease causing microorganisms. So that means also it is beneficial to the plant. Therefore, it ensures better plant growth and development. So this also acts as a biofertilizer to the plant because it provides a macronutrient, that is phosphorus, to the plant, and rhizobium provides nitrogen to the plant. Both nitrogen and phosphorus, they are like macronutrients, very, very important nutrients for plant growth and development. The third example is cyanobacteria, which are also called as blue-green algae. These bacteria are also called as blue-green algae. They also help in fixing atmospheric nitrogen, and thus they add organic matter to the soil. So some of the examples of cyanobacteria like Anabina, nostoc, they are examples of cyanobacteria which fix atmospheric nitrogen into the soil. So these are examples of some microorganisms which help to fix nitrogen or phosphorus into the soil and therefore act as fertilizers. But at the same time, they do not have any disadvantages of chemical fertilizers attached with them. Therefore, it is completely beneficial. So we get the benefit of fertilizers. At the same time, we do not have any risk to the environment. Now, we spoke about nitrogen fixation. Now, you might be wondering that how is this atmospheric nitrogen coming to the soil? Because we know that atmosphere, the air which we see in the atmosphere, that contains 78% nitrogen. So 78.3 or 78, approximately we can consider it as 78%. So 78% of nitrogen is present in the atmosphere. Now this percentage remains same all the time. Now when we say that these microorganisms like the rhizobium bacteria or the cyanobacteria, they fix atmospheric nitrogen into the soil, so does that mean that they take some nitrogen from the atmosphere and fix them into the soil? If that is the case, in that case, this percentage of nitrogen in the atmosphere should decrease, right? Logically, if I say that these microorganisms are taking nitrogen from the atmosphere and they're fixing that nitrogen into the soil, that means the percentage of nitrogen in the atmosphere should decrease. But it says that the amount of nitrogen in the atmosphere always remain constant, and we all know that. As a matter of fact, that the amount of nitrogen in the atmosphere is always 78%. Have you ever heard that it is sometimes 42% or sometimes 30%? No, it is always 78%. So how is that percentage maintained always? However, we know that there are organisms which are also fixing nitrogen into the soil. So that sounds quite interesting, that there is no decrease in the amount of nitrogen, but at the same time some nitrogen is getting fixed in the soil. So this entire thing is driven by nitrogen cycle. So nitrogen is constantly being exchanged between different components of the earth like atmosphere and soil through various organisms, and this cycle is taking place continuously. As a result, the amount of nitrogen in the atmosphere always remain constant. Like if there are some organisms which are taking up nitrogen from atmosphere and fixing them into the soil, at the same time there are some other organisms which exist which convert the nitrogenous compounds present in the soil and convert and send it back to the atmosphere. So both type of organisms exist. So some of them are taking in nitrogen from atmosphere and fixing to the soil. Some of them are taking nitrogen from the soil and sending it back to the atmosphere. So as a result of these two processes taking place simultaneously, the amount of nitrogen in the atmosphere always remain constant. So now we will talk about the nitrogen cycle. We will try to understand the concept of nitrogen cycle. So this picture tells everything on its own. So I really don't need to explain much, but still I'll just try to explain you how exactly the amount or how exactly the nitrogen balance is maintained in the atmosphere. In fact, not only atmosphere, but the balance is maintained between atmosphere and the soil. So let us see how nitrogen gets exchanged between various components of earth. Now, as we all know that the amount of nitrogen which is present in the atmosphere is 78%. Now, there are certain organisms like the nitrogen fixing bacteria which undergo the process of nitrogen fixation. For example, rhizobium is one example. Cyanobacteria like Anabaena, nostoc, they are another examples. So all these bacteria, for example, rhizobium or nostoc, Anabaena. So here I have shown in the figure rhizobium which are present in the root nodules of leguminous plants. So these type of bacteria, they convert the atmospheric nitrogen and fix them into the soil in the form of ammonia. So now this ammonia in turn, ammonia is a nitrogen containing compound. So this ammonia in turn can be utilized by other plants for their growth and development, because nitrogen is again a very important constituent or is a very important nutrient which is needed by the plants. So these plants will utilize the nitrogen which is present in the soil. So that's how plant will get its share of nutrients. Now these plants in turn will be eaten up by another animals, maybe the herbivores. So the herbivores will get their share of nitrogen. These herbivores in turn will be eaten up by the carnivores, so they will get their share of nitrogen. So that's how nitrogen will be passed on from one organism to another. So now all these organisms contain some nitrogen in some or the other form. Now what happens when these plants or animals die? Now when the plants and these animals die, then what happens? Now the dead plants and animal remains also contain a lot of nitrogen. So what happens to this nitrogen? Now what do we do with plants and animals which die? We bury them under the soil. So once they reach the soil, so basically nitrogenous wastes reach the soil. So they are in the form of nitrogenous wastes. That is, they are waste materials which came out of dead and decaying matter, and they are rich in nitrogen. Now these nitrogenous wastes are then acted upon by decomposers. And who are decomposers? Primarily bacteria and fungi. They act as decomposers. That is, they break down the organic dead and decaying matter into simple inorganic form. So these decomposing bacteria, they will convert it into simple inorganic form, that is nitrogen, and these nitrogenous compounds will be present in the soil and they will again get utilized by these kind of plants. So that's what will happen to some part of the nitrogenous waste. Remaining part of the nitrogenous waste will be taken up by the denitrifying bacteria. Now what do these bacteria do? They will convert the nitrates back into atmospheric nitrogen and return it back to the atmosphere. So if you look at this entire cycle, what do you see? On one hand, nitrogen is being taken out from the atmosphere and fixed into the soil. On the other hand, nitrogen is taken from the soil and given back to the atmosphere. So this is how the amount of nitrogen in the atmosphere always remains constant. And also if you see amongst the various living organisms, the nitrogen continuously gets exchanged, or it continuously passes on from one organism to another. So this describes the nitrogen cycle, and this helps you to understand how exactly the process of nitrogen fixation takes place. So let us quickly have a recap of whatever we have studied in the previous slide, that is nitrogen cycle. So we know that atmosphere has 78% of nitrogen gas. Now this nitrogen is fixed into the soil by the nitrogen fixing bacteria. One example is Rhizobium. So what do these bacteria do? They convert atmospheric nitrogen into nitrogen compounds like ammonia, and these compounds remain in the soil. Now when they remain in the soil, plants utilize nitrogen from the soil through these compounds through their roots. Now the plants get their share of nitrogen. Animals eat plants either directly or indirectly. So animals also get nitrogen. Now when these plants and animals die, their dead bodies, they reach the soil. So therefore the dead bodies contain a lot of nitrogen. So nitrogenous wastes reach the soil. Now there are two types of organisms which will act on the nitrogenous waste. One is the decomposers, which will convert some part of this nitrogenous waste into nitrogen compounds, which will be further utilized by the plants from the soil. Whereas other type of bacteria will be denitrifying bacteria, which will convert the nitrogenous wastes into atmospheric nitrogen and send it back to the atmosphere. So roughly this is how the nitrogen cycle takes place. So now let us look at how microbes are so good as decomposers. So just now I touched upon the decomposers, saying that they help to decompose the dead and decaying matter. So let us see how exactly they do it and which are the microbes which act as decomposers. So what is decomposition? Composition means formation. Now we often say, right, this is composed of this and this. That means this is made up of. So composition is always about making something. Decomposition is just the opposite of that. That means breaking something. So this is the process in which complex organic matter are broken down into simpler forms. So what is this complex organic matter? The complex organic matter is nothing but the dead remains of plants and animals. So that is because they are all organic, they are carbon containing substances. So that complex organic matter will be broken down into simpler forms. Simpler forms could be inorganic materials, maybe nitrogen, phosphorus, which act as nutrients for the plants. So it could be inorganic nutrients, or it could be inorganic substances like nitrogen gas, which is released back to the atmosphere. So basically complex organic matter are converted into simple inorganic substances. So the organisms which help in this process of breakdown, they are called decomposers. So the names have to match. Organisms which help in decomposition has to be decomposers. So bacteria and fungi are the important decomposers. So basically only these two classes of microorganisms, they act as decomposers. So in a way we can say that these decomposers help us to clean the earth. Had these decomposers not been there, in that case the dead plants and dead animals would have just accumulated on earth, because there would have been nothing else who could have broken down the organic matter into these inorganic substances, and these inorganic substances eventually get utilized by the plants. So they help environment to get rid of dead and decaying matter, as I said just now. Otherwise half or more than half of the earth would have been dumped just by dead bodies. Enrich the soil quality because it converts it into inorganic substances like nitrogen, which definitely improves the quality of the soil and also its fertility. So the process is really simple. Complex things are being broken down into simpler forms. So the picture itself speaks for itself. You have something very complex made up of a lot of things. You break it down into simple forms. And these simple forms can then be utilized by some or the other organism existing on the earth. So it is not causing any sort of environmental pollution in any way. And who helps us in doing that? Bacteria and fungi. So end of the day, they are again the microbes. So see, by now we have actually seen microbes doing so many good things for us, for our environment. So we cannot say that microbes can only cause disease and only cause harm to us. So there are so many friendly microbes as well.
So what do you guys say? Was the video useful? So if you have a feedback to share with us, do let us know in the comment section. And I will see you all very soon with a new video, with a new topic. Till then, take care. Bye-bye.